So satellite cells are normally quiescent, so they are in a resting phase until they are activated. And so once they're activated, it's like to replace damaged cells, they become activated and they can kind of differentiate into a new muscle fiber that eventually will fuse with the existing muscle fiber. So they're kind of a replenishing cell that only gets activated under certain conditions. Otherwise, they're just kind of waiting. And so there's a lot of research going on kind of what those signals are. So besides damage, what will induce a satellite muscle cell to self-renew to itself, and which ones are the signals that cause it to differentiate and become more mature. Okay, so this is important if you're studying diseases that affect muscle, like Parkinson's disease, or if you have some nerve-related damage to your muscle and you want to regenerate muscle cells. So the satellite cell and identifying all the players that are important for its self-renewal and differentiation capabilities are an area of active research. And they're another example of a stem cell that exists in the adult. Okay, so that's the last lecture. So today, what we're going to talk about is we're going to pick up again talking about induced pluripotency. But what we're going to do first is talk about talk about some other methods that have been used to reprogram cells in the past that show plasticity of gene expression. And we'll talk about stem cell therapies as a medical implication of developmental biology, and we'll pick up our discussion of comparing embryonic stem cells with induced pluripotent stem cells. Okay, so we're going to compare and contrast the different methods that have been used to reprogram somatic cells to a different cell fate or a different type. And for here, we're going to talk about somatic cell nuclear transfer. We're going to talk about transdifferentiation, and we're talking about cell fusion and IPS using viruses. Okay, so we've talked about plasticity and flexibility in certain cell fates, but gene expression is also plastic. So plasticity can refer to the ability of committed cells to change their genetic program. And there are several lines of experimental evidence that show that gene expression is indeed plastic. So nuclear transplantation and cloning experiments, cell fusion experiments, transdifferentiation experiments, and IPS cells. We're going to talk about each of these types of experimental evidence today. Okay, so some simple experiments that have been done in the frog show that the nuclei of differentiated cells can support development. And so the experiment that was done is basically transfer of a nucleus from a adult skin cell or a mature somatic cell into the enucleated cell from a tadpole. So here they took an adult frog, they cultured the skin cells, and they took a tadpole and took epithelial cells from the tadpole. And what they did was they irradiated the adult cells to basically destroy the DNA in the nucleus of those cells. This is the next experiment. Okay, so basically this is combining two experiments in one. So you take the nucleus from the adult, you take the nucleus from the tadpole. Right, so you're taking the nucleus from an adult environment and a more younger environment. And they transfer the nucleus into an oocyte that has had its nucleus removed. you're putting in a more mature nucleus into an oocyte cytoplasm. And you transfer the nucleus in, and what happens is, in both cases, is that tadpoles develop from this oocyte that has the nucleus transferred into it. So this means that there's something in the cytoplasm of this egg that has told the mature nucleus and the tadpole nucleus to develop tadpole-specific genes or development-specific genes. The other example of reprogramming is called somatic cell nuclear transfer, or SCNT. So in this case, a blastula was taken from a frog, and nuclei from individual cells was used. And so each of the nuclei in this blastula should contain the same genome, right? They're coming from the same organism. They should express the same DNA. And so what they did was they took those nuclei and transplanted them into egg cells that were enucleated, similar as before. And what they get from these individual oocytes are identical frogs. So you have cloned frogs that express the same genetic material. Okay, so what's interesting, okay, so let's take a step back. So this means that, again, this was more differentiated in the blastula stage. So it should have been, these cells would express genes that are more likely to be involved in differentiation to the blastula stage. The oocyte is just an oocyte. And so it has, and the nuclei of those oocytes were removed and replaced with these more mature temporal stage nucleus. And what they got was full development of identical frogs. If you do the same experiment with adult cells, they're less successful. Okay, so that says again that something in the adult nucleus is being prohibitive for somatic nuclear transfer, but it can still happen. Okay, so this shows that the genes that are required for early development are not irreversibly altered, so they can be reactivated if given the right environment. And it's cytoplasmic factors in the egg cell that are important for regulating gene expression in the nuclei after transplant. So we know that there are a lot of cytoplasmic factors that can affect, say, cell division, like cyclins, and etc. But there are other cytoplasmic factors that can also regulate other genes that can cause, basically, reprogramming back to a more embryonic-like state. And the ultimate example of this is Dolly, the sheep, where this is the first time that somatic cell nuclear transfer was shown in a mammal. So it's not a specific feature of non-mammalian cells. So in the case of somatic nuclear transfer in the sheep, oocytes were removed from a donor, and the nucleus and the spindle were removed by basically sticking a very small pipette into the egg and removing it. And then more somatic cells were taken from another sheep, and they were used as the donor for the nucleus. So these were taken from the udder of this sheep. The nucleus was isolated, and then the nucleus. I'm sorry. Here they took cells. Okay, so here they took the oocytes, and they took the whole cell, and they physically fused these cells together. And what you got was basically the egg that was fused with the udder cell that was a mature somatic cell.